Friends, now populations become a little bit thin. It's okay. No, there are some features I could see. It's fine. Maybe they have better business than attending this. Also, that's fine. After reviewing broadly about the genomics and genome projects globally, and what they did, what they were going to assess from the genomic studies. So they were going to quantify the genomic variations. Let's quantify or understand briefly about genomic variations. So my outline of the talk is about genomic variation broadly, types of variations, and our own work on copy number variations when we did in our lab, and COVID-19 and its variations, where a couple of days back, weeks back, I updated myself that okay, it's a very interesting one. I'll talk about that and conclusions. I'm talking about somebody has asked the questions. These two individuals, can you put the light of at least few lights off? Some of the images I am not able to see. So the figure there. Some lights. Yeah, it's better. No, for them also. I don't know. Can you see these images? Because very rarely see this kind of individuals. Very, very rare. This is the very rarest one. Two individuals, Sultan and he. They are very different. You can see that. What is responsible for that? One is 8.1 feet and another 2.5 inches. What is the differences? Quick. You had, everybody has this vada, right? Yeah, then speak. What is the difference? What could be the differences for the different insights? Genetics. Right? What kind of genetics? Genetics is broader. It's a phenotypic variation. Let us take only one character here, height. Height variations. His height is controlled by a single gene, or many genes, or many factors. Three options I give. Is it controlled by a single gene, many gene, or many factors? Which one is right? First, second, third. Many factors. Not only gene, I told you in the beginning of my talk, genes and environment also more important. So the majority is a gene, but environment also contributing factors. Here, the height gene is showing. Yes, many factors means many genes and yes, their environment also responsible. Now, if you go and somebody wanted to work on project work, some young faculties are here and he will help you. If you showed all the height genes and the variation height gene in the global genome, you can make a nice review article and nice which is responsible for this kind of the phenotype and this kind of height. It's easy to do that. Available in the database and literature. This is because of variation in the height genes. Because of variation in the height genes, they are different height. They are normal. Generally, genetically they are normal, but they differ in the height. So that is the changes in the genome sequence of this individual made them from a different height. This is one of the examples. Globally, including all the ethnic group, already I told you 99.6% we are all identical, 0.4% we are different, indicating that 4 million bases within two individuals. This is the range I am talking about. If you look at the two individuals, maybe between these two individuals there will be 30 million, 10 millions also. If we pull all our individuals here, 30 million variation total you can report. That I'm talking about the single nucleotide polymorphism. Between two individuals, at least on an average 4 million. If you pool whatever the variation I and myself or Dr. Sripada has, may not be the same variation, may not be found within any two of you. So that way all the variation good. So the data tells us about more than 80 million variants are there in genome or 3.2 billion base sequences. Pooling all the global population. Don't take only only 80 million variations. Where to compare all the global population, a pool, the present data tells that. But between two individuals, minimum 4 million variations. What are these variations? When you said this, who is normal here? Your registrar, vice chancellor, because they are the top people of the university, or our Sidramaya ji, or our Modi ji, or our president. Who is normal here? Or the HOD, your HOD, or me, or anyone of who is normal here? How do you quantify normal? With the genome data. This question is clear?
Yes.
Then what is the answer? Can you predict? We are all normal. How much? 99.6% we are normal. Broadly 0.4% we are not abnormal. We are having variation. Don't say abnormal. We are normal. 99%, 99.6% our the nucleotides are identical. That's what happens. Okay. There are two terminologies to understand the genomic variation. We should understand what is mutation. Very good. So whenever I go to and ask question, sudden change is a mutation, they used to say, a student, because in PUC textbook, some of our Mahanabhav, they written the same, the variation carried for it. How come sudden come? It's a heritable change. Heritable change is called as a mutation. Okay? It's not a sudden change. Suddenly it will not. There are sudden changes in our somatic cells. They are there that will end up with a cancer. So sudden, it's not a sudden, it's because of the defect of different factors will be. So mutation is a change, heritable change in our genome. How frequent it is? We should know the basic, basic information we should know. What is the mutation rate in generally, spontaneous mutation rate? Somebody taught you or some textbook, did you see that? Then somebody was talking here, I told them also. It's very rare. 1 in 1 million to 10 million. Out of 1 million nucleotide, 1 nucleotide is very change. Or from range 1 million to 10 million, that range is the mutation. Very, very rare. Mutations are not very common. Okay? So, this is called spontaneous mutation. This has been worked out in bacteria, in Escherichia coli. That's the one in 1940s, and when they started using chemical mutagenesis, radiation mutagenesis, they worked out this is a mutation. Spontaneous mutation, very, very rare. This is a rare. 1 in 1 million to 10 million. So depending on the situation, what kind of the culture they use and so on. That's broadly. Okay? But you see the changes are occurring there. What is the definition for polymorphism? Earlier said the single nucleotide polymorphism, SMP. Everybody knows that. What is that? Single nucleotide polymorphism. How frequent it is? What is that single nucleotide polymorphism? Is it a mutation or different? Is it a mutation or different? Broadly. So let me put it, the variation. Let me put the term variation. Variation means any change of the nucleotide in the genome is the variations. What is the difference between variation and mutation?
Heritable.
Heritable, rare change we call as a mutation, but other changes or polymorphism. How polymorphism? Very frequent variations are polymorphism. So we generally classified that around 1% in the population, out of 1% in the population. That means 1 out of 100 or 1 in the population are more frequent, 1 out of 100, the variation what we find is single nuclear polymorphism. Single nuclear polymorphism also change in the nucleotide. That can also call the variation or the mutation. But we cannot call them in a true sense because the earlier people have coined that term mutation is a rare phenomenon. I'll come to that mutation. Mutation is a variant. It's also variant. Polymorphism is a variant. Mutations are very rare, less than 1%. Polymorphism are more than 1%. Out of 100 individuals or 100 nucleotide, 1 variations we see. After 100 nucleotide, 2 variations we see. Those are polymorphism and the mutation. So we have to distinguish them very, very clearly. Any change is called variation, and the mutation, polymorphism also variation. Depending on the frequency of occurrence of these variations, we call them as mutation or the polymorphism. I hope it's clear now. Now I am talking about genomic variation, including mutation and the polymorphism. So genomic variations increased in population by 3 phenomena. One is the mutation, the changes, certain rate changes in the population, that because in geographic area, that environment, or in the chemical induced area, different, or naturally during recombination, replication jitter or replication error will be there, so that mutations are occurring there. That's one. Sexual reproduction in meiosis and fertilization. Meiosis, parental gametes were undergone reduction, division, undergo recombination. There also exchange, the combinations are there. The recombination introduce the variations. And during fertilization, egg and the sperm and egg DNA undergo recombinations. There also the recombination introduces variation. And after that, inbreeding. What is inbreeding? I don't know in Mangalore, but at least in Karnataka or Andhra, uncle-niece marriages are more. Because we don't want to give a property. My ancestor made the 1,000 acres of the property. I don't want to give to somebody else. I want it to be my uncle or my niece. There only my property only. Take the one. Along with that, they gave also, along to the family, genetic variation, more genomic variations. That is, inbred population contributes more of deleterious recessive variations in the gene. Okay, my last sentence, I wanted to all of you to read that. Non-lethal mutations or variations are, sometimes the mutations or variation, and I am using that, you consider as a common there. Or reservoirs of genetic diseases. What does it mean? If the mutations or the variation occurs, that gene is defective, the cell cannot survive and the organism dies there only, lethal. Okay? Only the variations are there, but it's not affecting the affecting the survivability, but they are making the organism survive with defect in one organ, defect in the reproduction, defect in lung, or genetic defects are there. So, non-lethal mutations are reservoir of genetic diseases. Whatever genetic diseases we have, for the survival there is not a problem, but they have different kinds of mutations. I hope to understand. And that's very, very important. If it is lethal, a dominant mutation we have in Drosophila, curly, dominant, the gene is present in second chromosome. If they come as homozygous, the embryo will not form at all. Homozygous dominant lethal, it will not be. Only in heterozygous we can maintain that aspect. That too, the balance, the manner. genomic variations are introduced every generation because the meiosis, because of the fertilization, because of gametic segregation there, and because of inbreeding introduces, and deletes already told you, changes are very, very rare. Lethal changes are very rare, but you are not able to record them. And removed by selection pressure. What is selection pressure? They are not able to survive, they are removed. The embryonic abortion. So they conceived, she conceived, but within 2 months there is nothing. Or within 2 weeks, gone. Because of this lethal or translocation which are not able to sustain and proceed them. Often this alteration, the altered signaling pathway within and between the cell in their physiological development, these variations. So signaling pathway, any of the signaling pathway, it has to communicate, it's not communicating. One of the dyslexia gene called protopedaline, gene is present on X chromosome, I don't know, I might have a slide there, but still. It's a adhesion protein, one cell to another cell adhesion protein. If that protein is not holding these neurons together and the communication, dyslexia. Remember Tare Zameen Par movie? Anybody has seen that movie?
Yes.
And they cannot able to communicate properly in the beginning. That's the problem, communication problem. Because this gene is defective. One of the genes, this is defective. I'm talking about signaling. Sometimes the genomic variation which encode the proteins are often pleiotropic or loss of function. They have multiple function or they do not have any function. So genomic variants are inherited in patterns, different kinds of patterns. So classification of genomic variation, I already told you, genomic variation or mutation they cause, like spontaneous or induced. Spontaneous is very rare. Induced, we can use radiation, chemicals, or our transposons like you use in Drosophila, the P elements and transposon maize, they use transposons or in insertion sequences in bacteria, they can cause that also. By site, mutation can be classified. Tumor cells, they become somatic cells, they become tumor. Germ cells, they inherit it, or any region of the genome that can be expressed. By function, revertant, lethal, loss of function, gain of functions. If some mutations are there, gain of function, the regulatory region, the function is enunciated, like oncogene, proto-oncogene become oncogene, tumor suppressor gene, loss of function by mutation. So that goes. And by fitness, neutral, beneficial, harmful. The wolf, which is there in Arctic region, it's survive better. If that bring it to the Mangalore here, how long it can survive in this environment? Because that the hair is problem there. It's harmful for its survival in this environment. And the size SMP or the segment or whole chromosome is deleted or added there. Why you have to study the genomic variation? To understand that the variation is neutral or pathogenic, or to understand where they localize, the size, and what kind of variation to cause the phenotype. And we also use this genomic variation, copy number variation for human migration, and in population plasma, people have appreciated that also. It obeys or it supports the work of Lalji and our Pangarash work of the mitochondrial and Y chromosome data from out of Africa, and what we made little bit changes there. I think I might have there. Okay, a second settlement in China, and gender discussion also based on the copy number variations. So that I think I'll skip this copy number variation here. The copy number variation, 4 types: SMP, single nuclear polymorphism, and microsatellites. The microsatellites, I hope you know DNA profiling. 4 nucleotide or 3 nucleotide are added and deleted that will make the variation in the genome. And 100 to 1000 nucleotides are added, deleted, they are called as indels, small indels. And 1000 nucleotide to the 5 MB, 5 million nucleotides are added or deleted. They are called copy number variation. Deletions, insertions, or duplication. They can duplicate it also. 3 category copy number variation, which occupy larger space in the genome there. Above that we have classical thing what we teach the students. The chromosome variation, what are those? Deletion, duplication, inversion, and translocations at the chromosome level, submicroscopic level. Okay? Now what I am talking about, within the genome where you cannot see it there also. So 4 to 24 MB due to the genome copy number variation, about 2.5 million base pair due to SMP, single nucleotide polymorphism in the genome, that is the total genome, some article has quantified that also. So I already told you about 6% of the humanity. So let me talk about this 4 genomic variation very briefly. SMPs, as you already know, the single nucleotide change. Any change in that, 90% of the genomic variations have this SMP. And here look at the cytosine here. 2 out of 3 SMPs are replacing cytosine by the time. Okay, about say 60% is the kind of replacement here is the cytosine, the SMPs there. Totally about in the human genome, about 10 to 30 million SMP pulling all the individuals the variation. SMP can occur any regions of the genome, coding or non-coding regions, and we do have SMP consortium in NCBI the database or any of the EBI database or Japan data DD the database. You can see the consortium, whenever a new SMPs are identified, they add it to the answer. So I hope you know this, the transition and transversions. What are transition? Purine to purine, pyrimidine to pyrimidine, or transversions, vice versa. They make the change of 3 different kinds of mutations. Silent mutations, where the cysteine becomes cysteine, though the change is there in the 3rd position. And missense mutations, the 3rd position change is there, but here this called missense or nonsense mutation, non-synonymous mutation, whereas the cysteine becomes tryptophan. Where nonsense mutation, the 3rd position cysteine becomes the stop codon there. So what kind of variation, what kind of changes are there depending on the functionality, the synonymous, non-synonymous, or loss of function changes. So already I told you this example, I'll skip this. This is one of the image what my project students now also they do. I'll give some gene, I'll find out the variations in the text mining and also in the databases, pool all the variations and map one particular gene here. What is the MET gene is involved in congenital heart defect. One of the last year, Bhumika, she just I wanted to make it also. And now here how many E stands for exons, I stands for introns there, and there's 21 exons are there, where exactly the mutations are there, what are the mutations repeated, what are the end of the stop codon. So I'll make them to map here. Then we'll start to look into that, where exactly, then we will go to the protein of the same gene, protein domain, which domain the mutations are there. So the domain is very important. If it is interlinking, and which variation is very dangerous, deleterious. We have prediction tool, apply the prediction tool to confirm that out of 1000 variations, these 3 variations are very deleterious because they frequently occur in the populations, and this is the deleterious one. And there are so many variations. Earlier I said the only 3, now missense, synonymous, non-coding deletion, non-coding insertion, in-frame deletion, within the frame deletions, and stop gain variants, frame shift deletions, frame shift. One nucleotide is deleted, the frame is shifted, and the coding property of the amino acids vary, the protein is not functional there. So no, the protein is something different. And the two in-frame insertion, all these are the one causing the variations in the genome. Talking about maybe now. I already made a difference between the SMP and the mutations. SMP, once it occurs, it is there in the population, it is there in the generation. So these individuals have, these are the SMPs inherited. This many the lineages, in these lineages, one generation, second generation, third, fourth, fifth generation, they inherited. Mutations very rare, only one here, mutations here, rare here, they inherited. So this is the one. So we carry with the haplotype from our parents, grandparents for 10 generations we carry, because that haplotype regions do not undergo recombinations. The same block is carried. So that way we can trace population specific, caste specific variation in running in the population, and that way we can make a good treatment for this individual. We have minor allele and major allele. Generally, major alleles are the one who more frequently occurring. Minors are the rare allele, they say, and we have to look at the major and minor allele, and rare allele we have to look at for the rare genetic diseases. That way, the blocks will go to P1, P2, the 4 individuals, and this is the one. So we can classify them also. This is more important, and that's called haplotype block. When I say haplotype block, that the gray block. Within the gray block, whatever the variations were there, they carry generation after generations. 90% all variations are common variations, and very rare variation generally cause rare diseases. So people are looking for rare variations, rare diseases, and variation for rare diseases for the treatments. Many of the genomic studies they are looking for that. But common variants already there, we can also, people are looking for to make a suitable drug for these common variations. So I complete one aspect of broadly review the single nuclear polymorphism, the now microsatellites, essentially and extensively used for DNA fingerprinting, DNA profiling for the even plants and animal industries. And trinucleotide repeats are there. For example, Huntington's disease at the 4th chromosome, P16.3 region, CAG repeats up to this repeat, CAG repeats can tolerate up to 35, become normal. If it is more than 37, become Huntington gene is adding more of the repeat there, and the mental retardation, fragile X of the X chromosome, the CGG, if it is more than 200, as a mentally retarded, one X chromosome present in male, males are more effective, like other diseases. So this variation can occur in the coding regions. They can occur in the coding regions, non-coding regions. There are different diseases are associated, or by the 3' gene, or in the coding Huntington, as I told you, the reason also. So they are randomly distributed. Frame shift is one nucleotide. I hope you know that the whole message will be gone there. Frame shift mutations are more dangerous because the whole protein is there but is not functioning. So some of the, I don't think it's interested, some of my students have done data for the SMP analysis and single nucleotide indication of constantly heart defects, blood-borne, and this asthma, the IL-4 they did, and the insertion deletions also for my project students. All these are project students, MS project students are there, they publish that also. I will come to the 4th category, very important, the copy number variation, 1 kb to 4 several MBs, is with more than 90% identity between the genome of different individuals, and this is because of deletion, insertion, and duplications, and they are more deleterious. And this, how do they cause copy number variation? What are the mechanisms involved for copy number variation? when you say copy number, a segment of DNA is varying, deleted, added, and duplicated. The reason for is homologous repeats. Sometimes repeated sequences are there for recombination. The machinery confused with that, and it will add up that also. Segmented duplication, chromosomal rearrangement, duplicate transposition, the cut and paste of the transposition phenomena, or copy and paste the transposition. Non-allelic recombination occurs, and this generally should have been 2 copies here, like here. It sometimes deleted only 1 copy, here both the copy deleted, here 3 copy or 4 copy. This way, copy number variation present in our genome. Whereas, look at the Craig Venter genome, he has more deleted genome than this. So, I'll take 1 or 2 examples of our study, lab study, that copy number variation. We use the microarray 6.0, the SMP and CNV, and 1.8 million markers were there, and every markers are between 0.5 to 1 kb markers, and average of 680 bases. And I don't need to explain the techniques, it's a common technique. And this is the kind of the pipeline, the protocol we use to understand this. And the message, what we did in this. So we have studied 1715 individuals, only 38 individuals from our lab, subjected for microRNA 6.1, 6.0 array, and others are from the available from the database. HACPA sequence, 270 HACPA sequences here, and the 31 Tibetan samples. There are raw data available from EBA, European Biotechnology Informative, the database. We have the MOU and took the raw data. We analyzed the Chinese samples, 155, and 2 German samples, 2 samples, and Taiwan sample, Australian samples, and the new one that is from American samples. 12 populations of ranging 1,750. This work has been done by 6 or 7 of my PhD students. Now, this work was not their PhD work. Extra work, curiosity, they did from 2012 to 16 they did it. We have about 30 publications asking different questions, and also that has pledged me, allowing them some of the awards, this analysis. So we did the copy number variation as a genetic marker to trace the human evolution and as a marker for disease analysis. So this is the source of the genome from the Affirmative Genome Wide and the EBA, the accuracy got the data. And these are the different softwares the students learned, and the tutorial they taken, they did it. I don't think any our teachers taught us, even I may not be able to do all of them are also, because they learned and they did, but you monitor them also. So because I want the same thing to be done by 2 different students, because I have to internal validate, otherwise something goes in a publication, we will be in problem. So far we didn't have any problem in the publication, because many high level institute or papers are retracted, faculty are leaving the job in India, also top level people, and they are cheaters. So not university people, they won't do that. So I have not seen any other institute data. So this is our data, some simple data. These are the individuals belong to this whole population. Just look at some of the thing what I have marked here. African population here, they have about 3 point plus or minus here, the standard deviations here, about this many MB of copy number variation we found, and the China 3 point, another China 6 here, and look at the Japanese 3, and New World is American about 13, and whereas India is here, our data 8.9 data also. So to summarize that, more of duplication we are seeing in the total population than the deletion. 75% of the copy number variation what we recorded is a duplication and only 25% deletions. And the size was the smaller size, the deletion duplication more than the 5 MA deletions or duplications. And this took months together for my student. All this duplication they here, 44,109 CNV, they map for individual chromosome, all the 24 chromosome, they all individual here, some spots 12 population here. This population there, they mark that also. If you zoom it, you can get all those kind of information. And a leader for this Avinash Virappa, he took the lead and all others, 6-7 members work and gave worked, some person, the first author, second author, that way. So I divided all the 7 members for 7 first authors, and Avinash took 2 or 3 first authorship also there. So we had 44,000 global CNV identified, and also the population, the chromosome wise also we could able to, which chromosome has the more CNV, I quantified here, the more CNV is seen in 14th chromosome, look at here, 14th chromosome here, in all the population, 8th chromosome, and 2nd chromosome, and the 15th chromosome, where is the 15th chromosome here, see, but less CNV is at chromosome 13, 20, and 18 chromosomes. Population wise, identified the deletions and duplication, coding and non-coding region, it varies. One example here, that coding region duplications, here the dark green, look at the dark green, which are the population here. German population, African population, here the Tibetan population, Indian populations are having more of the duplication in the coding region, and here the New World also having that. Which is the more, here look at the Chinese population, China and Japan having more of the coding deletions there. So we again compile the student that let me compile the which are the copy number variation associated genes in different pathways, and also like development process, responsive stress, and the biosynthetic process, signaling cascade. So they map them also. Finally, we came up with the out of Africa that we published. So what is the message here is that from the 3% of the copy number variation we found in Africa. Second settlements about 6% in the China population. From there it dispersed to the other region, whereas North America here is about 16% of the copy number variation. Latest derivatives, this we use, and new population with the highest copy number variation we published in class 1 2015. It's 14. So summary, the copy number variation map we generated, what does it provide? A rationale for prioritizing chromosomal region. This chromosomal region is having copy number variation, and this we have to be very careful. If there is a mutation, it ends up with a population specific genomic variation, and uncovered the elements in the genome that had largely been unexplored here. The same copy number variation of 44,000 copy number variation, then we ask the question that diabetic susceptible gene, where they located, how many copy number variations are located in that, in this 1700. These are the normal individuals, so-called normal individuals. And does that map with the case studies also? We did it. Diabetic, I don't think I need to tell about that. Diabetic, that time I'm going to be more. Diabetic has many consequences here, and it's insulin resistance, and the global population about 10.5% or more. People are in India, they're trying, all the doctors say, oh, more are diabetics, and also there are many reasons, because major reason is that all the doctors, many of the scientists, many of the administration in India ignoring the genetic factor there. They just simply lifestyle, you ate the smoke, you ate this food, that also contribute, but genetic factors all these are ignorant, educated persons. I'm telling you about that. So they should know some analysis about genetics, the individual, the family, and so on. So, in diabetes, highest duplication seen in 8 population here, the one where the amplification means the duplication here, the darker one here, the German population, Chinese population, and Tibetan and Indian population duplication associated CNA in the Indian population, those duplicate associated for the diabetic gene, 82 diabetic genes. Hotspot we made, another, the software they use. Look at the 22nd chromosome. Okay. 22nd chromosome have more hotspot, more individuals were found the diabetic gene in this individual, then this hotspot, the others are less. And this also mapped onto the individual chromosomes. And none of the diabetic predicted genes are found in 9th chromosome, 21st chromosome and Y chromosome, and more of them are copy number associated diabetic gene were found in 22nd chromosome also, that region. And we also made the circus one comparing sharing of this. So 82 genes and 24 the genes identified in the common population. With the Australian case studies, they identified the hit in the diabetes. So Australian population identified the defective gene, there is 24 genes to defective. Those 24 genes were mapped to our out of the 82 genes here. That means whatever is suspected in the common population, the normal population, was also seen in the case studies. And also we identified hub for this diabetic 3, major hub for the diabetic. I think these are the E to F1, EGFR, F, also these are the one. And what we have identified the pink, wherever you have the pink, we identified that the red was the primary interactions and the blue type 2 diabetes not under daily burden identified. And also this is published. This work was the MSc project students, the Manasa provision, and we had a good report, and these 2 members were associated with that to guide and also as a mentor. 34.5% of the genome is under the CNA burden for the type 2 diabetes. 34% of the genome, the genes, that segment is involved. And we could establish population-specific genome organization. 82 disease-causing and associated genes were identified in this and indicating that copy number variation may play a significant role for type 2 diabetes. MicroRNA, also the same 44,000 genes were understood, and I will not go detail about it because they got the Nobel Prize here. I will only tell about this is history about this, how this microRNA identified, and we also mapped the microRNA gene in our 1,715 individuals. We identified 6,742 microRNA gene, and the microRNA associated microRNA gene associated with CNV, and target gene where the microRNA is there, they are also identified, and another the interacting protein for this microRNA there. So the 3 different targets were identified, the CNV, and this also published, I think Avinash, the first method. And dyslexia, another interesting one. Look at the pedigree from some of the students who can biotechnology or zoology students. What is the pedigree says here? The dark regions indicate that they are male and they are defective, right? The male. Grandfather, father, and 2 twins. Did you get that? Father, grandfather, you can see all. can't see here. I am putting my oh, that's the. I thought this also reflect there. No. It's difficult. Okay. So all this father, grandfather, and two twins. We found there, and also individual there mentioned that the copy number variation in all these four individuals in a protothedrin gene on the X chromosome. Okay. How much time I have? Oh, yeah. So, to make the story short, all this male, we consider that the Y chromosome has a mutation culprit gene, Y chromosome do not have the culprit gene there. But we did see in the X chromosome. How come the X chromosome, they are all male are affected? And to trace back to that, we identified that there are some undergo recombination between X and Y chromosome earlier to this, and the Y chromosome gene, a protothedrin gene, moved into the X chromosome, they undergo duplication, and that has caused disease. So, how come the Y chromosome and X chromosome undergo recombination? In 1980, there are two regions between the X and Y chromosome that they show the recombination. We call it pseudo-autosomal region 1 in the top and pseudo-autosomal region here 2. They are called, there are two regions undergo recombination between X and Y chromosome. This has been identified Goodfellow in 1980 in UK scientists. But now we identified another region here undergo recombinations between X and Y chromosome gene are moved to the X chromosome, and we proved behind out with PCR amplification direct and the in vitro studies, and that because of the Y chromosome mutated duplicated gene and moved to the X chromosome, mother, because she has a 2 X chromosome, and she has not shown that only the X chromosome, what the guy, they got it, all the males, and they show the protothedrin, and also this is the one we published after 2 years of the rigorous the studies, we did that. So we name that as part 3. Many of the scientists, Indian scientists, are skeptical about university scientists. No, I can't agree with that. After publication also, I sent this to all the top people of the country, even including CCND, my colleagues, and finally, Ramchandra is a good one. At least I got this one, even including Professor Sharachandra and Thelma and Raji and everybody wrote that about that. I wanted Partha Majumdar also, they said that. And they said, new discovery, if this has been done in some good institute, they would have got the Bhatta of that day itself. They are not recognized. Even later, skeptical. No, how can you say our Arun Kumar from IAC? No, Ramchandra, how can you say this? I got it, I have the evidence, etc. So you have to, yes, somebody has to do it. I don't have the other kind of tools to repeat it. Okay, fine. So my message here is that any observation, let it be the simple experiment, basic experiment, whatever you do in the laboratory, research scholars make an observation, write it down. Someday, some time, when you become faculty, you can answer that question. So, or you can discuss with like this in the conference, and you can get the resolution. So, I hope you know that I might have told you also. These are the two ATGC. Here, these two are the purines. Purines are what are the purines? Adenine and guanine have double ring. Okay, that's why the size is bigger. These two are the shorter. They are the pyrimidines. They have only one ring. There, yeah. If you hold like this, they cannot. If you turn like this, 20 angstrom from here to here, 20 angstrom, they can match. It is simple. This was resolved. One of the scientists, after the conference, evening, they were sitting here and they discussed, why do you problem about that? They just only, you reverse this. They said, he got an idea, they did it, the Watson, and even this idea, the Chargaff had that, would have resolved the issue 20 years earlier also. Okay? sometimes you have to go to the conference, discuss with them, the greater, this is the one. Even for the Sanger DDRC, German scientists were resolved that, no, I wanted to stop my polymerization for the complementary study set. What is the problem there? Remove one of the H there, it will not polymerize. The DDRC there, the OH is there, the 3' to polymerize. Oh, it's nice. The guy came out and called his lab, a postdoc in the lab, you synthesize the molecule, the DDFC molecule, and after coming back to the conference, I wanted to test it. After going back, already they had the results. So this is some of the example in the conference. This was told to me, one of the great scientists from Innsbruck. I was there for 1 month to teach genetics in the the Mendel's name, in Mendel's place. In Mendel's place, I went there to teach genetics, and there the Renaud Rieger, as a senior scientist, in his garden we were sitting and drinking in the evening. So don't think that I am a drinker, but after the mountain walk, he brought in myself and Dr. Hartstein and Peter Ludana. They were sitting there. This is the one. Do you know what happened? He used to tell that. So I mean, when you interact, you will get some good idea of that also. Even reading also you may not get it. These are some of our senior publications. COVID-19. COVID-19, I am not going to explain this. The 29,000 nucleotide it has, 29.9 kb sequences it has, and data from bad viruses, and it has a very interesting 2.3 of the genome of the ward of 1 and 1A and B, and 16 structural. What I wanted to tell is that because of genomic variation, different kind of genomic variation, they hit your Omicron and you have different kinds of genomic variation. They become alpha, beta, delta and gamma. These are become the called VOC, that is variant of concern. They made that all this kind of variation we can see here, but only the VOC variants of concerns are propagated in the sensitive individuals, and they fix there, and that has spread. That's why we had a second wave of more death. And what are the landscape of this pandemic? Here, genome, viral mutations, 1 in 1,000 mutations were there. This is the way. And host immunity, some of the hosts do not have the immune system, are very sensitive, and vaccinations, so vaccination, whatever the kind of vaccination is there, some little control about the viral replication, then the immunity is increased, and global, the present day book in the global, that is the matter. So I was curious to understand this, and I made that none of the geneticists are making in the news. I made a small article in Deccan Herald, and that is May 21st, 2021, and they published it. I sent it for 10 newspaper. Luckily, that is Deccan Herald people recognized my genetics view of that. I explained about how mutation occurs, how mutations are going to be fixed, and how they have been spread to the population also. This is available, and you can read. Okay, this is the one you can get it also of this. So every individual is different in their genotype. That makes some people die, some people are even with some medication, vaccinations or some whatever medication, they came from the hospital to home. So the pandemic to the endemic, because of many of the reasons, they could immunity, all these are become endemic. So more variations, because of immune system is there, it's not going to affect that will taking care of the dust pandemic to become endemic there. So population immunity occurred by vaccination. The COVID vaccination, or earlier had different kinds of viral bacterial vaccination, that also to some extent there. People will cry, no vaccination, vaccine was not been tested for 12 years and so on. In a crisis, they bypass certain things, but you need what? So let's not worry too much about the 1% and 2% or 5%, 10%. They have not followed that, but the same thing. humanity globally. Let's think in those directions. And if there is any mistake, let us correct it next. So do you think that while this made last month, now there's already new virus has come now. The COVID virus can be there in a populations. It depends on what new variants are there, how new variants are fixed in the susceptible individual, and how it propagated, propagated there, and that how strong it is. It can be there. So, not all the individuals, the whole global individuals in a remote island, remote places are not vaccinated. And even they have died, this vaccine is mutating there, the sensitive population, no one knows there. So they can also become a place for spreading that also. We cannot rule out that. I'm not telling that tomorrow itself we start wearing the mask here. We have to be open mind, these are the genetic phenomena. Susceptible, chronic infected, immunosuppressed individual might be there somewhere region. There can there in JN.1 the COVID virus has a 96% of dominant allele of changing that. If they found that might again change, because 1 in 1,000 they changes there. And this is the new now 2 COVID virus they infect one person, and there the immune cell is both undergo recombination, they produce a new one there. Hybrid one, what's called XEC has been produced a few days back. That is, this has been published last month, yes. So, variations can be found in both exons and intronic region. 95% of the genome does not code for the protein, and proper diagnosis or analysis of genomic variation is essential for human welfare. Proper diagnosis, I'm talking about diagnosis. Once you diagnose that you have this problem, there are n number of treatment, n number of drugs are available, related drugs are available, they can be given. My concern is that. So, this work was done by Dinesh, Ranjini, Kusuma, Avinash, Sangeeta, Radhika, Raviraj, Megamurthy, Somana. I guess these people are remarked for this work what I presented here. And I thank you.